In this video, we will discuss about the in vivo results of this MCL-1 inhibition and also this Dasatinib, which inhibits the proliferative genes or the genes that are responsible in the EMT, that is epithelial to mesenchymal transitions. So what they did is they took the cell lines that is derived from the patients who was affected with the pancreatic cancer and did the xenograft injections on the mice model and studied the mice model for 5 weeks. And what they are able to see that there is not much change in the cancers when they see using the immunostaining or bioluminescence, but and these things are quantified in the panel B also, where we can see that the total flux is in the end remains same for all the conditions, that is either they are treated with the Dasatinib or this MCL-1 inhibitions or the combinations. However, when they check the weight of the tumors, they are able to see that the weight of the tumors is low compared to the is low in the case where this dual treatment is used, that is where Dasatinib and MCL-1 inhibitions is used compared to the control. And same things they have observed for the proliferative markers, that is Ki-67. The decrease is small, but it is statistically significant. And when they check this CC3 positive area, they do not see too much difference in this. And when they check about the lungs, the pancreatic cancer spread in the lungs, they do not see any metastasis, or that minimum metastasis has been seen in these lungs. And these things are quantified in the panel H, where we can see that there is no change in the metastasis area in the vehicle. And in the case where treatment is either used by Dasatinib or MCL-1 inhibitions or the combinations, but when they check in the liver, they are able to see some of the suppressions in the metastasis when they use the Dasatinib or MCL-1 inhibitions or the combinations. And these things are quantified in the panel I, where we can see that the decrease in the metastasis area is maximum when the Dasatinib and MCL-1 inhibitions are used in combinations. And these things are complement by this immunostaining image also. So finally, what they are able to conclude is that when this MCL-1 inhibitions is used, or such as using the BH3 mimetics, then it will trigger upregulations of this MCL-1 that could be because of the survival mechanisms. Whereas when this SRC families inhibitors are used, it will trigger this upregulations of this SFK and coughlin, and that plays a role towards invasions. The relationship between this coughlin and SFK have been covered in our previous video. So in the end, finally what they are able to conclude that the in vivo results also, they are able to see some suppressions of these proliferative behaviors, that is shown by these Ki-67 markers and the metastasis area in the liver, that the combined MCL-1 inhibitions and Dasatinib has some synergistic effect, and that will lead to the reductions of the pancreatic tumors. And these things has been validated in the mice models where they are infected with the uh, cell lines which is derived from the patients who was affected with the pancreas. But one thing that we need to understand about this mice is that in these mice models that they have used for the study has their immune system compromised, or this immune system compromised mice has been used for their studies. So I hope you understood this their results and what they finally conclude in this figure. If you need to understand more about these experiments in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.